Okay, welcome to your Chapter 17 supplementary video. Of course, the title of this chapter is Environmental Hazards in Human Health. And as usual, you should be at the point of having read this chapter already and looked over some slides in your worksheet. Something I want to talk about that's not really covered much in your chapter reading is exposure to toxins. And I would just mention to you first three general routes of exposure. So first of all, you can have gastrointestinal. So if you ingest something, making sure I'm spelling this right, gastrointestinal, dermal, so on your skin, and then inhaling it, respiratory. So these are, if you will, exposures, how you get exposed. So those are pretty straightforward. And then there's also the types of toxins you can have. So you can have respiratory toxins. Oops, spelled that wrong. I think I might have mentioned this in the slide section. Respiratory toxins. We have metabolic. And then you have like neurotoxins. Neurotoxins. I can't spell today. And endocrine toxins. This is just in both cases. We're just naming some of the clear common ones that we come across. So now it's important to realize that if you are exposed from something by inhaling it, that would be a respiratory exposure. It doesn't necessarily mean it's a respiratory toxin. It's important what the mechanism is. So for instance, carbon monoxide is an example of one that is a respiratory exposure. You breathe it in, it's a gas, carbon monoxide, but it actually, the mechanism by which it causes problems, it is actually a metabolic toxin. So I just want to point that out. We're not going to go into all the details and mechanisms, but I do want to point out a few simple mechanisms to you about how some of these neurotoxins behave. It's just interesting, and it's good to get a feel for that and how they actually work. So one of the mechanisms I want to talk about, these would be metabolic toxins. I'm not going to talk about carbon monoxide. Carbon monoxide actually, what it does is it binds your hemoglobin. And hemoglobin, of course, is the component of your blood that transports oxygen. It binds your hemoglobin more effectively than oxygen does, so effectively you suffocate. That's why, obviously, if you start your car with your garage door closed, that's dangerous because you literally could suffocate. You basically just fall asleep. That's a metabolic toxin, even though it's coming in through the respiratory system. Well, the other ones I want to talk about here are some of your heavy metals that are whoops, I'm telling you, with my spelling today. Heavy metals is just kind of a term that people use for toxic metals that represent elements, and they're not always even metals. Some of them are metalloids that are kind of lower down in the periodic table, a little bit heavier than things like lithium and sodium and stuff like that. So some of the ones we see are mercury listed, lead, cadmium, and arsenic. Okay, so you can look at those on your periodic table and find them. Once again, mercury, lead, cadmium, and arsenic. And one mechanism by the way these exhibit a toxic effect is by binding enzymes. Now, enzymes are in your body, and they pretty much are specialized proteins. Okay, so what they do is they act as catalysts. And a catalyst is a substance that speeds up a reaction and isn't really used up. So that means it can speed up a reaction and then go and speed up another reaction. Now, in the next section that we're going to cover, atmosphere air pollution, I'm going to talk about catalytic converters. And I'm going to talk to you about how a catalytic converter speeds up a reaction. Well, same type of thing, only these are in your body. Most reactions in your body are sped up by catalysts. They just wouldn't happen fast enough for you to live. So it just speeds up a reaction, specialized protein. Okay, and they always work by binding a substrate. So a substrate is just a word for whatever its partners that it's going to bind in your body in order for it to speed up a reaction. So I'm going to draw you very simple. We're going to use the Pac-Man here as an example. Some of you might not know what the Pac-Man is, but it's not the greatest diagram. So this is my substrate, and the enzyme that comes along is going to look like this. And I'm just drawing something. So basically, we use this idea of a lock and a key. They fit together. And so that's the way that enzyme might be folded, or maybe some sort of functional group it has, group that's hanging off it, makes these two go together very well. It turns out that these enzymes in our bodies have a lot of sulfur associated with them. That's a bad color choice, sorry. Maybe go with like a green here. Have a bad color choice. Have a lot of sulfur. Turns out that these heavy metals, they like sulfur. And when we draw a conclusion like this in chemistry, it often applies to multiple systems. So if I have a beaker of mercury and I have sulfur, they're going to bind together. They like each other. If I have mercury in a lake and there's sulfur in the lake, they're going to bind together, and it turns out they're going to precipitate. They're not very soluble together. If I have sulfur in my soil and I have mercury coming through leaching, it's going to bind to the soil, to the sulfur in the soil. If I have mercury entering my body, it's going to find that sulfur in the enzymes and bind to it. And what it does when it binds to it, it kind of changes it. It changes that enzyme structure, and it no longer is now able to bind what it was supposed to bind and renders it ineffective. So that's one way in which these work. This is just one example of the mechanism. Another one I mentioned were these neurotoxins. And the neurotoxins have another mechanism of working. And here's an example of just one. So you have nerve cells in your body, right? And these are two different nerve cells. It's basically the end of one and the beginning of another. And we have neurotransmitters that move across these gaps. This is called your synaptic gap. If you've taken some biology, and the neurotransmitter moves across there, and it binds to like a little receptor here. And that's how information is transferred. The nerve impulses are transferred from one nerve cell to the other through your body. Turns out some of these neurotoxins, what they actually do is they'll kind of mess with this system in here. Sometimes they'll actually bind an enzyme that's responsible for breaking down the neurotransmitter, or they'll block the receptor here, things like that. And like for instance, some of these neurotoxins that are insecticides, they do this in the insects, and basically winds up with this uncontrolled nerve response, and that leads to like the insect having like paralysis and ultimately dying. So it's just to give you a couple ideas of how these things work, because it's really fascinating biochemistry. The next thing I want to talk about is this dose response curve. And this is covered in your book pretty good, and I want to talk about this, where we have these curves here now, and we're going to measure this by dosing animals. And on our x-axis, something we learned in our lab, is that this is going to be the independent variable, and the dependent variable is the response over here. So the dose of the chemical and the response that we see, and that response could be death, it could be lots of things, but one of the things is death. Okay, and you might see a curve that looks something like this. And what you're seeing here is there's a dosage that has no effect. And at some point that effect kicks in, and that's what we call our threshold. Okay? And there's a point we have 100% response where any additional dose doesn't do anything more. And this is in your um, slides, or it's in your book actually, in the chapter. Okay? So this is a dose response curve. Okay, and you can actually use curves like this to calculate what we call an LD50. So what you do is you say 50% of your response, where's that a call curve right here? You go over and you figure out, okay, what dose is that gives me a 50% response. Okay, that's how we use dose response curves. Now what I want to do is I want to show you a basic calculation here. And this is something that I don't stress a lot of calculations in a class like this because you guys aren't scientists, but it's good to brush up on your math skills. So we're going to read this now. An average cup of coffee contains about 100 milligrams of caffeine, and that's in, just a regular coffee as opposed to, espresso or something like that. Espresso or maybe a darker roast that has a higher caffeine. And the LD50 for mice and rats, which are typically two of the animals we use to dose to figure out what kind of effects we have, is 130 milligrams per kilogram. Now, assuming this is the same for humans, and this is what we do because we don't like to dose humans with chemicals, of course, ethical problems with that, so we dose animals. We want to figure out the number of cups of coffee that would ultimately be fatal to a group of adults that took this coffee all at once. And if let's say they're all 150 pounds. So what we're going to do first of all, we're going to convert because now you see, notice what we have. We have pounds here, but we have kilograms here. So I would give you a conversion like this if I expected you to do something like this on one of your quizzes. We have a 150-pound adult. What I'm going to do is I'm going to use a common conversion. You'll find 2.2 pounds is equal to 1 kilogram. So we can do it like this. And what this is allowing us to do is, this is our conversion factor. It allows us to get rid of pounds. If you remember your math, anything that's in the numerator and the denominator can be canceled out. It's kind of like saying, once again, bad choice of colors there. Kind of like saying, what's 1 over 5 times 5? Well, they can cancel those both out. It's equal to 1. So we've got rid of our pounds. We now have kilograms. And the answer winds up being here 68.2 kilograms. So effectively, that's the average weight of this group of people. Now what I can do is I can say I've got 68.2 kilograms as the person's weight, and I know the LD50 is 130 milligrams per kilogram, right? So this is going to tell me how many milligrams is going to kill 50% of the population. And that turns out to be 8860 when you do the math. 130 times 68.2. Once again, the kilograms has been canceled out. We're at milligrams. Now we know each cup of coffee has 100 milligrams, so we can figure out how many cups of coffee that is. And ultimately, I have 1 cup has 100 milligrams times 88. And I've written this down here, but that's obviously 88 over 1, so don't think that's in the denominator. I just wrote it low because I'm running out of space here. Milligrams, right? That's going to cancel out with this milligrams because that's in the numerator, this is the denominator. That's going to give me the number of cups of coffee. And you see now it's 88.6 cups of coffee. So you realize that caffeine is not a particularly toxic substance because that's a ridiculous amount of coffee. And if you're drinking 88 cups of coffee a day, you have more problems than worrying about caffeine as your LD50. So you could see not a particularly toxic substance. And I think I just left another blank there in case I needed the space. I didn't realize that. Okay, but we're good here. Now I want to talk about biomagnification of toxins. So one of the things we're seeing here is that certain chemicals actually collect in the fatty tissues. And I talked about this in the slides, and your book talks about it a little bit, not really, actually, very much at all. So fat-soluble toxins, they could collect in the fats of your body, and what then happens is they don't get metabolized when another organism eats them. So let's say we have DDT, which is an infamous insecticide that was used in the 60s and the 70s. And then it was was banned in the United States. We don't use this anymore. And it's because, and this all came out of Rachel Carson's Silent Spring and that whole era of the environmental movement, establishment of the EPA. So what happens here is that the DDT collects, say, in the small little plankton that are in the lake that the DDT runs off into after it's been sprayed to kill insects because it's a neurotoxin. And then little fish like minnows will eat a bunch of those little plankton, but they don't metabolize the DDT. It just collects into their tissues, and you get more and more and more. And so when you get to the top of the food chain, which is like the big fish and ultimately the birds of prey, and this is what happened, they wind up with huge concentrations in their body, which starts to have negative effects. And we saw this with the bald eagle and other birds of prey. Of course, the bald eagle became the symbol of that because that was our national symbol. And it's kind of embarrassing if your national symbol were to go extinct. So that's what kind of led to the whole DDT and the bald eagle being kind of the poster child. But what it did was DDT interfered with calcium metabolism. That wasn't its design mechanism. It's a neurotoxin. But a lot of these chemicals have side effects, other effects in the body. And it interfered with calcium metabolism. Calcium was critical to eggshells formation. Thinning of eggshells resulted in a reduction in the population of surviving birds of prey. And that's what happened. So what you see here in this figure here is the concentration of the DDT. ppm is just milligrams per liter. Don't have to worry about that. That's the concentration in the water. Milligrams per kilogram. Sorry, that's the concentration in the fish. And this is the age of the fish. This happens to be trout. And you can see the longer the trout lives, the greater the concentration of the DDT because it's feeding longer and longer and longer. And that's what created the problem. This is one of the reasons we phased out these chemicals because they didn't break down in the environment. They're persistent. I talked about that in the slides. And that created lots of problems. So that really concludes the video. I want to summarize and give you an idea of what you should get out of the material for this one, as always. Make sure you understand what risk assessment and risk management are, what the differences are between those. Types of hazards. Your book talks about biological. Look over that. I don't hold you responsible for that. I really want you to mainly focus on the chemical ones because this is a chemistry class. But your book does talk about infectious agents and stuff like that. And it's good to educate yourself there. But carcinogens, mutagens, neurotoxins, and this stands for hormonally active agents. Those are the endocrine disruptors. And then how we go about evaluating hazards. Understand what a dose response is, what those curves are, what an LD50 is, what a threshold is, and understand what that risk is ultimately, because what you'll realize as you read is that chemicals in our environment aren't nearly as big of a risk as some of the other things. They are a concern because we can't control our exposure to them if they're being used on foods that we eat and things like that. So it's very important, especially for people that live a healthy lifestyle, not wanting to be exposed to these types of chemicals. But the real risks in our lives, if you're a smoker or something like that, that's a much bigger risk than something like pesticide residues on your food. And then the last section, which isn't covered much in your book, and I want you to be aware of, is fate of toxins. And I cover this in some of the slides, and that includes this bioaccumulation and biomagnification. With that, that concludes the video.